look forward to the meetings in the afternoon. Um, the So when we think about sensory motor transformations, you think about the sense organs, right, that detect all of the information from the world, and there are sensory processing circuits that take this information and compact it, and this whole process we understand as sensation. And then the information from all of our different sensory organs and processing circuits are integrated by central circuits in the brain to create perception. And then other central circuits take this perception and combine it with, say, our motivation and so on to decide on a course of action. Now, this is a more formal view of the sensation, perception, action, processing. Now, this view that I presented here is a serial view of sensory transformation, sensory motor transformation, and it arose largely from work in psychology, much before we started to understand sort of the mechanistic basis of how our brain works. Sort of, it precedes our even knowledge of neurons and synapses as sort of the mechanistic basis for neuroscience, right? Now, particularly work in the last 50 years from lots of different angles, right, has suggested that not only is this view likely limited, it may actually not be the right starting point for our investigations into sensory motor transformations, right? And the view or the set of experiments that have influenced me a lot are the ones from Melvin Goodell and David Milner, when they investigated this patient, D. Fletcher, who had a serious lesion in her visual areas. And what you can see here is what D. Fletcher is trying to do here is trying to match the angle of this postcard to the slot on the that represents the mailbox, right? And you can see that she's really all over the place compared to the control subjects. However, if you ask her to do a different task, that is actually post this letter, right, she does as well as the control subjects. Now, what this sort of experiment tells you is that you can do without seeing, meaning you can have action without perception, right? So there are a whole series of such experiments which cast serious doubt on the serial model of sensory processing, sensory motor processing. And in fact, the D. Fletcher experiment tells you that you can have direct links between sensation and action. We know from studies in all the sensory system that there is top-down control over sensation. That is, if you take visual circuits, there's a lot of feedback from visual cortex back into LGN. If you take olfaction, there's a lot of feedback from piriform cortex back to the olfactory bulb. So we know that this arrow must be there, right? Top-down feedback, top-down control. If you take the case of action having an effect of sensation, obviously that's very well established. Again, if you think of vision, we are not just passively looking at the room like a camera, but rather scanning the room purposefully using saccades. Again, if you take the case of olfaction, there's the whole sniff cycle where the mice and other mammals, they change their sniff cycle in response to odor. So clearly, action affects sensation. And finally, there's lots of studies which show that action, our interaction with the world, is really important for perception, right? So overall, it makes sense to think of these three entities as highly coupled sort of integrated system that are co-dependent on each other, rather being in some kind of hierarchy from sensation to action. This view is further supported by all of the anatomical work, right? So here is one example again from vision. This is all of the visual nuclei in the brain. And you can see that they're all connected to each other, densely connected to each other. Now, the really important point is all of these lines represent reciprocal pathways. That is each line here is a bidirectional arrow, meaning that they're again connected to each other bidirectionally, right? Implying again, supporting this framework of looking at sensory motor transformations. All good?
The slides aren't advancing on
the
thing, so you might want to unshare and reshare.
Okay.
Are you on a Mac?
No, PC. So the other thing is it does not see two screens. Yeah, I did.
All right, we're good. I can see this slide here. You want to put in presentation. So
Moving?
No, it's not moving.
Maybe I should go into. I think I've done this before. All right. So essentially, don't think of this sensory motor transformation as serial processing, rather a coupled codependent system between sensation, action, and perception. And what I'm going to do today is try to understand sensory motor transformation in this framework using the olfactory system of the fly. The fly's olfactory organs are the antenna and the palp, which have olfactory receptor neurons. And we are going to activate these olfactory receptor neurons and explore its effect on locomotion. And we are going to see, parse these effects into fast effects, meaning effects that are immediate, that require current sensory activity, and perception, which does not require the present sensory activity. All right, so the way we are going to activate these olfactory neurons is using optogenetics, because that will allow us to have very precise control over the stimulus and allow us to measure the neural activity under the same conditions that we do our behavior. So here is how we are going to do this. We are going to use a genetic tool that many of you might be aware of called Crimson, which is a red light activated channel. We are going to have a fly in a circular arena. The fly is forced to walk in this arena because it's in between two plexiglass plates. The center of the arena has a constant concentration or constant intensity of light. And then as the fly walks around and goes inside, because its olfactory neurons are expressing crimson, they are going to get activated, and the fly will think that there is odor present. And you can see here is an example where a bunch of olfactory neurons are being activated using crimson, and you can see that before the light is on, the fly spends much of its time outside, and then in the presence of light, it's spending its time inside, thinking that there is an odor there. The advantage of using optogenetics in this essay is that because you know now the intensity of light, and you know where the tracks of the fly, you can convert this intensity into the stimulus as a function of time seen by this particular fly, and then play back the stimulus in an electrophysiological ray and record from the olfactory receptor neurons. Um, so that's how we record from those neurons, and you can get the neural activity. So what we have is the neural activity from the olfactory neurons and the behavior in a completely freely moving animal. And that's what we are going to use to tease apart the sensorimotor transformation. Now here we are activating the olfo-galfo is US crimson. This fly expresses crimson in a large population of olfactory neurons. Well, what we are going to do is focus on a subset of olfactory neurons that in a previous study we had shown are activated by a strong attractant for the fly called apple cider vinegar. And just to make this clear, again, just like mammalian system, you have olfactory neurons. Each olfactory neuron expresses a single or a small number of receptors out of a large family of receptors, which not only determine where or which glomerulus it will go to, but also which odors it will respond to. And in general, most odors will activate multiple receptors and therefore activate multiple glomeruli. Different odors will activate overlapping subset of ORNs. And in this particular case, these 7 ORNs that the numbers here refer to receptors, they are activated by apple cider vinegar. And this is what we are going to focus on. So to sum it all up, what we are basically going to do is we'll activate different combinations of these 7 ORN classes. So essentially we'll have flies of different genotypes expressing crimson in different populations of ORN, and they are going to explore this arena. So we'll have tracks or behavioral results downstream of singlets, doublets, triplets, and here, so those are the triplets, singlets, doublets, triplets, and then you have these classes which don't have anything to necessarily to do with apple cider vinegar. They're just activating a large set of olfactory neurons. So we'll have behavior downstream of these 13 combinations. Then we'll take the tracks of each fly and reconstruct the stimulus experienced by flies and measure the responses in an electrophysiological ray, and use that information to understand sensorimotor transformation. Specifically, we are going to ask 3 questions. We are going to ask with sort of what we think of antecedent questions, that is, how to model locomotion itself and the effect of ORNs on locomotion. So modeling locomotion and how olfactory neuron activation affects locomotion. In the second part, we are going to ask how does locomotion affect a fly's sensory response. So that's that arrow going from locomotion to ORN activation. And then finally, we are going to ask the question about how all of these arrows interact with each other, that is the sensorimotor transduction. All right, so let's start with the first question. How do you quantify a fly's locomotion and the effect of ORN activation, right? So these are sort of flies of the same genotype. You look at the green tracks here, you know, they look the same, or they look different, right? Yeah, we need some kind of answer for that, right? Are these tracks the same tracks, or are they different tracks, right? That is are all of these can be thought of in the different instantiation of the same stochastic process, or are they fundamentally different? And then the other question is, you know, at least for 3 of these flies, it's pretty clear that the fly is spending more time inside when the ORNs are stimulated, right? Now the question there is, what is the exact difference between the green and red tracks there, right? Or what is the behavioral algorithm used by the fly, and how does ORNs affect it?
I'm super sorry, but I missed the critical point
Yeah.
back in the beginning. So why, when the light goes on or is it activated, do they fluoresce in the center?
Why do they do that? Because inside, when they go inside, the ORLs are activated.
Oh, I
And-
see. I got
Yeah, because they're expressing red light activated channels in there. And so we are going to ask these two questions. And the fundamental approach of asking this question is we measure parameters from the empirical flies and create a generative model. That is, we make synthetic flies and we ask, do the synthetic flies look like empirical flies, right? And let me describe this process in the next few slides. So the model I'm using today is an agent-based model. This agent starts with walk. And the reason we call this walk a curve walk is because there's enough curvature even when the fly is trying to walk straight that we have to account for it. From the curve walk, it can go into sharp turn where it turns really sharply, or it can stop. And again, a stop and turn because the flies, when they walk and then they stop at a position, the next time they might move in a different direction. Therefore, it's a stop and a turn. And then you have the boundary condition because the behavior of the fly, the boundary is quite different from its behavior in the center of the arena. So you have 4 states. And these 4 states are shown here. So boundary is green is those curved walks, and you have the red are the sharp turns, and then you have the stops in black. Now, importantly, each of the states in our model is modeled based on average states. Average curvature, duration is anyway one number. Curvature and speed are averages. So what that means is we can take this empirical fly, which can do whatever it wants, and substitute it with an agent, and you can see that the tracks which are modeled upon the averages are quite similar to the actual fly, meaning that the agent is a good model for the locomotion of the fly. This is the overall error for all of our tracks, which is about 3 millimeters, which is about a fly length. So what that means is that this is a good generative model for locomotion. Or in other words, what the agent, the power of the agent is, it can go sequentially from one of these 4 states. And all it can do is at the beginning of the state, choose its speed, curvature, and duration from an underlying distribution, right? So this is the distribution before the stimulation comes on, and every time it changes state, it makes a choice out of this distribution. Now, when you add odors, the effect of odor in this model is essentially to change the distribution, right? So this is an example here. And you can see that the curve of speed changes and the curve of duration changes, curvature remains more or less similar. Now, the other thing that ORN activation does is, because you have the stimulation zone in the middle, it creates an inhomogeneity. Let me show you what that means. Is that as the fly exits this zone, when the stimulus is present, it knows that something happened, and then it has the propensity to turn back inside, right? And this propensity is not captured by the kinematic aspects of the model that I was talking about, which can only allow you to change the distributions. And this is shown here. So that's the empirical fly. You can see that the turn density, which we refer to as decision density, increases as the light is falling, or the stimulus is falling down, right? Which is not captured in the kinematic model of the fly. So to incorporate this aspect of the fly behavior, we include a parameter called the border choice, which describes its increased propensity to turn. Now, the other thing is the turns. Question?
So the only changes are in the distribution, but not in the transition, the like transition probability between states. Is that true?
It will change by the virtue of the fact that the duration is changing. So if you decrease duration, you
Okay.
yeah, it will be.
Okay.
The other thing is at the border here, it has a turn bias. So usually the turns are unbiased. It would turn either way, but at the border, it turns in a way that this theta2, which is the new heading direction, forms a smaller angle with the radial vector than it was before the turn. So there is a strong turn bias when the fly exits the arena. And you can see that these two parameters that we call decisional parameters or directional parameters, if you want, they are basically you can capture those parameters by essentially applying them to the first two turns, right? The first two turns after it exits the central region, it has both an increased propensity to turn right after it exits. Compare the solid line to the dotted line. You can see that the later turns are do not happen at the border. The other thing is that there is this turn bias. You can see the first two turns are almost 100% invert bias, whereas the later turns are at chance level, right? So what we do then is we take all of these parameters, right? The kinematic parameters and decisional parameters that we measure from the empirical flies, and we then feed into our model, right? Obviously, remember, the empirical fly might be doing everything else that we are not measuring, right? So what this is telling you is, what this testing is, are the things that we measure, are they the important things? Are they enough to explain the behavior of the fly, right? And that's why you create synthetic flies. And what you see here is, you can focus on these guys. These are the full synthetic flies, and you can see that they capture the attraction index or the time that the fly spends inside. So that's empirical, that's the synthetic, they're quite similar, as well as the radial distribution of the flies of both the empirical and the kinematic flies. The kinematic plus decisional flies is the same. So the upshot of all of this is that we have now described, solved the first problem, that is how to model locomotion and the effect of ORN activation on locomotion. In summary, a four-state locomotor model where each state is described by average speed and curvature is enough. And then ORN activation has two effects. It changes the underlying distribution from which the fly is picking, and then it also causes an increased border choice and turn bias. So that's part one. Yeah, go ahead.
Yeah, can you clarify, please, the data that you presented? Is it with regards to any combination of ORMs activated, or is it specific to a certain, ORM? Do we always see attractiveness, or do we also see getting away from the?
We always see attractiveness, but the level of the changes and the underlying behavior is very different for different combinations of the ORM. This model, this basic model holds for every combination. It's not a different model for different ORM, but the extent to which different ORM activation are going to change speed and curvature is going to be very different for different orders. And that's what we are going to look at length in this third part. All right, but before that, we have to look into what locomotion does to the fly's sensory experience. Well, sensory experience. So the reason you generate a complex sensory experience here is because although the stimulus itself is static, right? This red light that turns on at 3 minutes and remains on for the duration. So there's no change in the stimulus itself. What changes is the fact that the fly goes in and out at will, and therefore it's going to experience a very dynamic stimulus environment just through its locomotion. And this is unique, right? Every fly has a unique sensory experience here just because of its locomotion. And therefore, it has a different ORN spiking pattern. So in other words, what we think of sensory experience here is this. What's the pattern of ORN activation for different flies? And as I mentioned earlier, the way we get at this problem is by taking these patterns of light and stimulating the flies in electrophysiological rig and recording, and you can see these are the responses, raster plots and the average responses at the bottom. Now, because we have 13 genotypes, 30 flies per genotypes, several minutes of recording per fly, that would mean a lot of recording. And frankly, it's unnecessary because what we can do is, after doing a bunch of recordings, we can essentially create this neural encoder, right? Which is essentially a series of 2 linear filters. Other people have designed such neural encoders and they work well, they work very well for us. And so using a combination of audio recording and this neural encoder, we have the responses for any arbitrary track that we produce. If you're interested, those are the filters that we needed. Now let's look at the ORN response in a little more detail. I'm going to present two tracks from the same fly, and what you can see here is the fly is outside, and then right at the border there's a stutter step there which pauses for a few seconds. You can see it here. So that's the light intensity experienced by this fly. You go from 0 and you just get inside, but not quite, and you have a plateau there, then followed by this large increase after that. If you look at the ORN response, in black here, again, an increase in firing rate, plateau, and then this large increase as the fly goes actually inside, right? Now here is a different view of this, where we are just plotting the change in firing rate as a function of time, and we'll see in a minute why this is important. Now here is another track. This time the fly just goes right in, and so what you see is a much sharper increase in response, which is much easier to see in this delta firing rate curve. So this rate of increase is much higher than the rate of increase there. And then the other thing is, after the odor comes off, in either case, you have this inhibitory period before it returns to the baseline. And this inhibitory period causes strong interactions, as you can see in the second and the third traces here, where the deltas are much weaker because of interactions from the previous pulses. So the upshot of it is that locomotion, when it interacts with the active properties of the ORNs, creates responses that are quite different from one entry to the next entry. And therefore, firing rate by itself is not a thing that you want to base your decisions on, because that's going to vary a lot. And it turns out that if you consider both the firing rate and delta firing rate, you're going to do much better. And this is illustrated here. So the horizontal line here is baseline firing rate. The flies start here. And then this is the first sort of entry when it stops here right at the border. And then you have the larger peak, larger increase in firing rate after the first initial pause. So that T1 corresponds to that large increase here. Now you can see that the responses are quite different for different flies. Now, in the ideal world, the fly would be able to keep track of its trajectory in this ORN space, but that's computationally quite challenging. So what it can really get away with is just keeping track of its firing rate and delta firing rate, instantaneous firing rate and delta firing rate. Because essentially, although the trajectories are different, it's going to follow the same, approximately the same pattern, which will become clearer in the next slide here. So what you have in the top here is sort of random sampling of data in the state space consisting of firing rate and delta firing rate. And what basically what's going to happen is as the fly enters inside, it goes through A, B, C, D, and E. So that pattern is going to remain consistent, even though there is a lot of variability around it. And you can see that here it enters A, B, C, D, E. That pattern is going to hold. And one can, if you keep track of your, if you base decisions just on F and DF, you will know where you are with respect to when you entered the arena. And this is probably becomes clearer in these color maps. So what we are plotting here is when the firing rate is in region A, where is the fly? So it's basically right at the border because the firing rate is increasing. Then it goes inside. In region B, it's mostly inside. In region C, it is again at the border because it's exiting the arena. In region D is the inhibition, and you can see that much of the inhibition area, it's outside, then it's not being, not even in the stimulus region, and region E is a low firing rate, and finally you have baseline. Now, what you can see here is that if you just rely on firing rate, you at the same position, you could have widely different firing rate. But if you rely on both firing rate and delta firing rate, you will have a better idea of whether you are entering the region or exiting that region. So the bottom line here in terms of the effect of locomotion is that the instantaneous F delta F pair serves itself as a short-term memory that marks its time from the time it entered the arena, the stimulus zone. So to sum that part up, right? So bottom line is the firing rates are highly ambiguous. You could be at the same location, and based on your previous history, you could have high firing rate or high delta firing rate. However, if you choose both F and delta F, it forms, because it forms this part of a similar trajectory across different entries, it marks, essentially it marks the sensory experience since the last entry. All right, so that's part two. And now let's look at basically the sensory motor transformation. So what's the sensory motor transformation? In the last part, I told you that a sensory motor, if you can always use more complex strategy, but if you are basing decisions based on instantaneous experiences, you will do really well, much better with F and delta F pair, right? So sensory experience, instantaneous sensory experience in our model is described by this F and delta F pair at a given time. And the behavior is described by this model, and what the odors or activation are doing is changing these distributions. And so sensory motor transformation then boils down to relating this F delta F pair to these distributions, which is what we are going to do next. So what we did, and I can go into detail later, but essentially we evaluate how the F delta F during the preceding 200 milliseconds before the fly takes a state decision affects the distribution of different variables. So what we are plotting here is just the mean for one variable, which is curved walk speed, right? And what you can see is that clearly at a given firing rate, draw a horizontal line here. The curved walk speed is not dependent on F, but on a combination of F and delta F, which I've already made a case makes a lot of sense. Now, so that's just one variable. There are about 9 variables in our data set, and these are the F and delta F mapping for different variables, and you can see that they are generally different for different variables. We don't have to go into the pattern of each of them, but I want to make one critical point, right? The sensorimotor transformation has basically 2 properties. First, the variable choice is dependent on F and delta F combination, and second, it's distinct from these distinct parameters. Now, why does that make sense? It makes sense, and that can be illustrated with these 2 examples here, right? So, so curved walk speed is small when the fly is inside, right? When it has essentially reached the highest stimulus intensity, it makes sense to decrease the speed and do a thorough search of that area. In contrast, when the firing rate or the delta firing rate is high, the fly wants to go up the concentration gradient or the light gradient, and therefore it's going fast, right? Out at the border as it's as it's entering the center of the arena. In contrast, if you think about sharp turn curvature, it makes sense to have very sharp turns as you are exiting the arena, as you see here. This red here represents very high turn curvatures, and what that's going to do is, as the fly is exiting out, is going to bring the fly back in, right? So really, that's sort of the magic of how sort of brain works. You have this parallel sensorimotor loop that somehow together makes sense by automatically, continuously adapting the motor program with very little information, right? Still instantaneous information. All right, so that all was for a single genotype. We have done such analysis for these 13 genotypes here, and I won't be able to go into all the details, but let me make a couple of important points. First is that for any given motor variable, in this case, oops, what is this? This is sharp turn curvature, and what you can see is that it's different for different genotypes. And the other thing to notice here is that when you have these single genotypes being active, there is not much effect on the sharp turn curvature, but when you activate them together, there's a much stronger effect on the sharp turn curvature. And by the way, I don't think I mentioned earlier, the reason we lose this flank sometimes is basically analysis, nothing to do with the behavior itself. We have very strict criteria of what points we include here. So if you don't have enough point at some data points, we just exclude it out. But you can obviously compare where there are points in each of these data sets, right? Now, you don't just see nonlinear summation all the time. In this case, there's not much nonlinear summation. And then if you add, if you add another ORN class to it, you have triplets, then you start seeing much larger effects. All right, so so that's one point, that is each motor parameters is affected differently by different patterns of ORN activation, and the rules of connectivity also depend on which exact ORN you're talking about. Then the other really important point is that this sensorimotor mapping is plastic. So the mapping that I've been showing you so far has been the average across the whole 3 minutes of when the fly is activated, when the fly is seeing the stimulus. If you look at individual time points, what you can see is that the increase caused by the high firing rate in this pattern of ORN is not present at 0 seconds, increases to 30 seconds, and then to 60 seconds, and then starts decreasing again. So the mapping from F delta F to different motor variables is plastic, right? Now, to sum this part up, The effect of ORN locomotion is essentially mapping from the ORN responses that is described by F and delta F to these different locomotor parameters. Then a given pattern of active ORNs that is a set, active set, just say OR42B, will affect different motor parameters differently. Rules of integration are diverse, and that the mapping is dynamic. That it's the mapping from F delta F to the locomotor parameters changes with time, right? So let me summarize all of this in this context, right? So what you have, if you take the fly's point of view, what it's doing is it's just exploring this area, and it's going to enter, it's going to make entrance into the stimulus zone, and at some point it's going to exit, and then again it's going to roam, and then again enter and exit, right? So it has multiple entry and exits into the arena. But there are two kinds of information it's getting, right? The first kind of information is short term, right? Every time it enters, the F and delta F function starts changing based on its locomotion. And that creates this trajectory in the F delta F space that I was talking about. Now, every time it exits, after some time, after a few seconds, it will completely reset. And so this fast information, right? These fast effects that last for a single entry is what it's doing to take these instantaneous decisions, right? Now, and it makes sense for it to evolve its decision as the F and delta F changes. Now, once it exits, although the ORN firing rate has quit, obviously its sensory experience still remains in some way, right? And it should leverage that sensory experiences. And the way it leverages that sensory experiences is by making this F and delta F map plastic, right? And so that its motor strategy then adapts over multiple entries into the array. Okay, so that's the crux of what we found. Now, how can this computation be done with neural circuits? Now, it turns out this is a fairly simple computation for a neural circuit. If you consider the fact that, I'm sorry for the hand-drawn diagrams, you know what I was doing last night. The idea is, if you take this ORN firing rate and pass it through parallel circuits that represent different filters, and if you differentiate, you will get a set of neurons that is active in A. Untransformed will be active everywhere. If you invert, it will be active in C. And if you choose integrators, it will be most active in E, right? So essentially, you take one and sort of branch it into parallel circuits that perform different computation, and then you connect it to these different temporal features, right? Now, it turns out that the Drosophila antennal lobe is well designed to do this. So this is just a different representative of the antennal lobe. In an earlier diagram, I talked about the ORNs. So now I'm talking about the projection neuron. Now it turns out that there are 4 classes of projection neurons. There are the uniglomerular projection neurons and multiglomerular projection neurons, and both of them come in excitatory and inhibitory phases. And it's entirely likely that these 4 classes of PNs carry these parallel information that can be integrated by these higher brain regions. And so we hypothesize that these PNs, as well as the neurons in lateral horn that are integrating these information, can perform the F delta F mapping that we require from one entry within one entry, right? And then you also have connections to mushroom body. Mushroom body only gets these excitatory PNs and not the inhibitory ones, and they can sort of make the map plastic. So they feed back. They also synapse on premotor neurons, and so essentially this connection takes care of long-term sort of experiences or those connections, right? All right, so let me step back from fly olfaction and tell you that what I've presented here is sort of very common for any sensory motor integration. So what happens is you have sensory input, and you calculate different sensory features from that sensory input. Many features are encoded by parallel circuits. Some features are important for perception. You take them and you create a model of the world. Others are used to drive a parallel set of actions. And then actions together lead to behavior, and its consequences are also fed back into perception, as are the set of actions. Now, perception can then reshape your next set of actions. And this general schematic is true for every sensory motor transformation, I would argue. And in the specific case that we see today, this set of actions is a probabilistic locomotor model with several variables, and this parallel sort of circuits are modulating these sensory features, and then using sort of sensory features that are calculated, say, at the antennal lobe or lateral horn or a combination thereof. And the same computation that you see here is happening, say, in the retina. You have 20 sort of sensory features being sent back to the brain, some of them important for perception, others for action. All of the work I've presented today is done by this single graduate student. He's really an outstanding graduate student. This is the rest of my lab, collaborators and funding. If you're interested in such work, I'm hiring. All right, that's it. Thank you so much. Go ahead, Jim.
All right, so you said you always see attraction, no matter what you're doing. Is that because you picked? So first of all, I'm surprised that you see attraction when you have ortho here then. So I assume that all of them have, I don't know if it's novelty or what, right? But, and then is the other attraction because you are pulling apart apple cider vinegar colorants, and if you instead used colorants that respond to negative valence odors, it would change? Or is it, again, a novelty thing instead of a, I'm curious how that works.
Right. So, yes. So the ortho attraction is, but we're not supposed people to this. You see it everywhere. And now what's okay? So we were really surprised by it. Okay. So what we did is this, right? We said, all right, at some point it's going to break, right? So we added another one, IR8. No, it's more attractive, right? So now here's I can speculate on that, right? What really it means? Okay, so let me in terms of attraction, right? So some of all of these guys, right? If we activate them, we'll see some effect on some locomotor barrier, right? And most of those effects is going to, okay, let me actually have this. I was going to, it's a better image. I forgot that I had this. Yeah, so that's your attractions, and you can see this ortho IR8A is much more attractive even than ortho, right? So those are your singlets, right? And all except for this guy, you can see some mild tendency to be near the odor, right? Now, when you increase it to number 2 of them, again, there's some increase, overall there's an increase in attraction. At 3, you're almost getting to the ortho level. And then this guy is somewhat less attracted even than 3. So I think the way we can summarize all this is that single ORN classes, by and large, are going to cause attraction in this particular arena. And the reason is that most of these ORN classes by themselves, they cause a decrease in firing rate in that inside region, right? Sorry, not firing rate, speed, decrease in speed, and actually followed with the increasing curvature when they get inside, right? So that essentially what it says is that when you activate most ORN classes, it's kind of when you're meeting, you're talking about the bad smell in the garbage, right? The first reaction to any odor, particularly these flies are hungry, is explore that area. How do you explore that area? You decrease your speed and increase your turn curvature. So you basically are in that area, right? And that's going to be dominant. That does not mean that there's no ORN that's not going to do something different.
And have you tried specifically to use an ORN that is activated by a negative valence odor?
The only one we have done is the carbon dioxide neuron, and yeah, I think that's inhibitory, but it's really funny. Once it smells the carbon dioxide, it either just exits the arena and sits there and does not move at all for the duration of the experiment. So it's very hard to get statistics out of that, but it's potentially repulsive. Questions? Yeah, go ahead.
So you use this paradigm to be very helpful.
Yeah.
Which then would be very nice to also add very clear boundaries. And that formation also helps disambiguate at the outside.
Yeah.
But we'll have to add a slowly changing radius.
Yeah.
The gradient on while it was present. But that begins to ambiguate the outside.
Right. So, great question. And the way we are working on this is, and I can show you the apparatus when you go to the when we have you over to the lab. Is we built a much bigger arena, this big, where we are actually following the fly in real time. And because we are following the fly in real time, we can give it any arbitrary pattern that we want. And that's one of the experiments we are doing, is doing a gentle gradient into the center, and we'll find out what happens, right? So I don't know yet, but it's a I think two things really is the F and delta framework may not hold, right? But what will hold is that from entry to exit, it has the circuit will have some dynamics, and it's going to act on those dynamics basically. And what the true nature of those dynamics are, we don't know. That's what we have to find out, is exactly what it's doing, is whether where the true limits lie.
I think we actually are at time, so we have to close.
All right, yeah.
Thank you so much.
Sorry.